In 2020, saying the virus came from China could get you removed from a platform. No argument, just straight up banned. Extreme cases of questioning the narrative had people lose their jobs, caused fissures between family members, or some were actually just publicly ostracized. The claim got treated as a statement about race, and the people making that argument weren't fringe. They were editors, fact checkers, and medical commentators writing in the largest papers in the country. A CNN medical analyst warned in the Washington Post that investigating the lab leak question risked provoking harm against Asian communities. Facebook straight up blocked the sharing of the column that raised it. Newspapers called it a conspiracy theory and in some cases tied the question itself to just racism. 6 years later, something very different is happening with the same fact. Peptides are now described as dangerous because they're made in China. That sentence gets published and everyone accepts it as the pejorative that it is meant to be. Nobody calls it what it is, a dog whistle. It shows up on the biggest health podcast where Chinese origin is treated as an assumed negative that needs no defending. The geography that was unspeakable in 2020 is damning evidence in 2026. Now, the obvious move here is to say one side is hypocritical, but it's not that simple. The people pushing the China origin argument about peptides today are not necessarily the same people who suppressed it in 2020. Country of origin isn't functioning as a fact in either case. When the fact pointed at a foreign government, it got turned down. When it points at compounding pharmacies competing with pharmaceutical company, it gets turned up. Nobody's principles changed, and that is most likely because the ad buyers remained the same, but they were buying a different narrative. So when someone tells you a substance is dangerous because of where it was made, they've told you nothing about the substance. They've told you who they're arguing with. Here's what makes this uncomfortable instead of just tribal. The quality problem with gray market peptides is real. It's worse than most people selling them will even admit. It just has nothing to do with the type of fear they are handing you. I'm going to spend most of this making the case that a lot of people should not be injecting these things, and I'm going to do it without once using where they came from as the reason. So here's where I stand. Before I go any further, I have a financial interest in a company called Alchemist. It's a functional diagnostics practice. It has full medical authority to prescribe, and peptides are one of the things that it can prescribe. My wife, Erin, runs the diagnostic side of it. So take everything I'm about to say with that in front of you rather than burying it at the end. Financial interest is a problem that influences ideas and advice. And so I want to be clear about mine. I also want to preface this. This is not advice. Nothing in this video is medical advice. I'm not a physician. I'm not diagnosing anybody. I'm not treating anyone. And I'm not telling you to start or stop anything. Those decisions belong with a licensed prescriber who knows your history. The clearest illustration of how little the word legal tells you about the word safe is happening in federal court right now. And it comes down to one molecule that isn't technically an amino acid. In September 2024, Eli Lilly sued the FDA over how retatrutide gets classified. Retatrutide is their next generation obesity drug, the one being talked about as a major step past what's on the market now. Here's the regulation. To be a protein and therefore a biologic, a molecule has to be an alpha amino acid polymer greater than 40 amino acids in size. Retatrutide is built on a backbone of 39 alpha amino acids. Attached to that is a single short chain containing two more pieces. One is gamma glutamate, which is an alpha amino acid. The other is a compound called ADO, which is an amino acid, but it is not an alpha amino acid. So count everything and you get 41. Count only the alpha amino acids and you get 40. 41 is greater than 40. 40 is not greater than 40. The FDA counts only alpha amino acids, lands on 40 and classifies retatrutide as a drug. Lilly says count all of them, lands on 41 and says it's a biologic. That's it. That's the entire case. The entire dispute is whether one molecule that isn't technically an alpha amino acid gets included in the court. In September of 2025, a federal court split the difference. It agreed retatrutide isn't a protein under the bright line test, but it set aside the finding that it wasn't analogous to a protein and sent that question question back to the agency. Lilly appealed to the Seventh Circuit and has said publicly it intends to file as a biologic regardless, with the target submission of the first quarter of 2027. So why fight this hard over accounting dispute? A biologics license carries 12 years of market exclusivity. A new drug application carries 5. That's 7 years. And there's a second clock nobody talks about. Under the Inflation Reduction Act, small molecule drugs become eligible for Medicare price negotiation 9 years after the approval. Biologics get 13. So winning biologic status doesn't just buy 7 more years of exclusivity, it buys 4 more years before the federal government can start negotiating the price down at all. And this is the one that matters for you. A biologic cannot be compounded. To put a number on why anyone is bothering, Mounjaro and Zepbound, the 2 drugs retatrutide is designed to succeed, brought Lilly $36.5 billion in 2025. 1 year for one company. So the classification decides 3 things at once: how long Lilly owns the molecule, how long before the government can touch the price, and whether anybody else is allowed to make a version of it. Nobody in that case is arguing retatrutide is more dangerous as a drug than as a biologic. It's the identical molecule either way. The phase 3 data doesn't change. The mechanism doesn't change. What changes is who's allowed to sell it and for how long. That is not a safety determination. That is pure profitability. And before anyone decides I'm on team compounder, this runs both directions. A compounding pharmacy has sued Lilly and Novo Nordisk for its federal court in Texas, alleging a coordinated effort to suppress competition and limit access to compounded GLP-1s. Everybody in this fight has a P&L and nobody in it is a practitioner, which is why is it legal is close to a worthless question to ask about your own body. And here is how fresh this whole battle is, because it moved a week before I even recorded this. On August 12th, 2026, Lilly filed 6 federal lawsuits against companies it accuses of selling black market retatrutide. 4 of the 6 defendants are Peptide storefronts by name. The other 2 are a compounding pharmacy and a med spa. The legal theory, which is the part you need to understand, is that Lilly is going after the phrase research use only. The allegation is that when a vendor labels a product research use only and then sells it without a prescription to people who are obviously going to inject it, that label is a false statement. Lilly is asking for permanent injunctions and disgorgement of profits, is flanking its competition in case the appeal falls through. They didn't lead with patent infringement. They went with consumer protection and unfair competition, which keeps the fight on the vendor's conduct instead of putting Lilly's own patents at risk in front of a judge. The lawsuits are the small part. Lilly says it has referred more than 200 individuals and entities to the FDA, to professional licensing boards, and to law enforcement. It says it has flagged more than 14,000 websites, ads, social media posts, and product listings across more than 100 countries. That's not litigation. That's an enforcement apparatus protecting its future income. However, what they are going after in the form of shady websites selling research-only substances is a real problem. These people also don't have your health in mind. A CBS News investigation in June found that more than 120 websites selling or promoting retatrutide and at least 50 clinics staffed by licensed physicians and nurse practitioners openly advertising it. After reporters made contact, at least 21 clinics pulled it or changed their language. One physician quoted essentially said, "Approval looked inevitable, so why wait?" And the harm data is not nothing. Human exposure to retatrutide reported to U.S. poison centers averaged about 95 cases a month in the first 4 months in 2026. That's 265% jump from the previous 4 months. Separately, the FDA reported receiving more than 1,700 adverse events reports tied to compounded semaglutide and tirzepatide as of May of this year. So when Lilly says the black market is dangerous, they're not making it up. They just aren't doing all of this for humanitarian purposes. So keep all of this in mind as we go into the most current events regarding peptides. In July, a federal advisory committee voted voted to recommend broader access to certain peptides. In August, the manufacturer with the most to lose filed 6 lawsuits and referred 200 entities to regulators and licensing boards. Same month, same molecule class, the opposite direction. Retatrutide is the one they're fighting hardest over, and retatrutide is the exact molecule in that amino acid dispute I just walked you through. So the company arguing in one courtroom that the molecule is a biologic that cannot be compounded is, in another set of courtrooms, suing the people compounding it. Those two efforts are consistent with each other. They're just not consistent with the story that this is about safety at all, because a molecule doesn't become safer when one company owns it. And to be fair, an unapproved drug from an uninspected facility is a real risk. And Lilly's chief medical officer is not wrong when he says what's on the black market is unverified. Both those things can be true. The risk is real and the enforcement is commercially motivated. You don't have to pick, but you can now understand the influence of what you are being told to believe. Now, if you think I'm reading too much into one company's legal strategy, let me show you the version of this we've already run all the way to the end. It just took 50 years to reveal itself. Psilocybin, the compound in what most people call magic mushrooms, has no reliably established lethal dose, or an LD50, in humans. The animal data puts the toxic threshold so far above any dose anyone actually takes that a fatal overdose is essentially unheard of in the literature. That does not make it harmless. Psychological risk is real and it's serious for people with certain psychiatric histories, but it means the physical danger and the legal status were never in the same conversation. LSD is one of the most studied compounds of the 20th century. Before it was scheduled, there were roughly 1,000 published papers and tens of thousands of patients who had received it in clinical settings. This was not just a fringe substance that nobody understood. It was one of the best documented compounds in psychiatry. Both landed in Schedule 1, which is defined as high potential for abuse, no accepted medical use, and no accepted safety, even under medical supervision. That scheduling wasn't a toxicology finding, it was a social one. These substances were associated with young people questioning authority in the 50s and 60s, with refusing a war, with walking away from institutions and back towards nature. Everyone, including myself, was under the impression that acid melted your brain. The generation those drugs got associated with, the anti-war generation, the thing they were protesting, produced a diagnosis. Post-traumatic stress disorder became a formal diagnosis in 1980, driven substantially by the Vietnam vets. 50 years later, the substances that were banned partly for their association with that war are being studied as treatments for the injury that that war and many wars after it produced. A Stanford-led study of the U.S. Special Operations veterans published in Nature Medicine in 2024 reported reductions in PTSD, depression, anxiety, and functional disability after a single ibogaine treatment protocol. We sent young men to do something they were not trained for, at least psychologically. It broke a number of them, and one of the more promising things we have found for putting them back together is a class of compounds we made a felony, in part because of who was using it while objecting to sending them. In April of this year, an executive order directed federal agencies to accelerate psychedelic research. There's a ARPAH funding attached. The FDA has granted breakthrough therapies designations, ibogaine, and IND clearance. And in July, 3 months after that order, Eli Lilly announced it was acquiring a company called Beckley for about $2.8 billion up front and with another $1 billion in milestone payments. The lead asset in that deal is BPL-003. It's a nasal spray. It's a synthetic 5-MeO DMT for treatment-resistant depression. It has breakthrough therapy designation, and it's entering phase 3. The pipeline behind it includes a DMT film and an MDA analog. This is the substance discovered from smoking the secretions of a toad. And this should surprise no one. Some of the milestone payments in that deal are contingent on the DEA rescheduling. It's the same company, the one in the amino acid dispute, the one that filed 6 lawsuits 8 days ago. 50 years of Schedule 1, then a policy shift, and within 90 days, $2.8 billion acquisition of the synthetic version with a payout structured around the scheduling itself. What I'm saying is narrower than that. The molecule has not changed. Psilocybin is the same compound it was in 1968. 5-MeO DMT is the same compound it was when it was discovered in 1936. What changed is that somebody figured out how to own a version of it. That's the pattern. Legality tracks ownership. It does not track danger, which is exactly what you're watching happen with peptides right now on a compressed timeline. It's the same mechanism, it's just a different molecule. So the regulatory picture moved twice this year, and it's getting misreported in both directions. In April, the FDA republished its interim 503A bulk substances list and announced it would remove 12 peptide bulk drug substances from Category 2. Category 2 is the bin labeled significant safety concerns, but it did not move them into Category 1, which is the approved bin. They are in limbo. Compoundable and prescribable, but not by the definition regulated. In July, the Pharmacy Compounding Advisory Committee met and voted to recommend adding certain peptides to the 503A list. The names under discussion included BPC, TB500, KPV, Samax, among others. More are scheduled for review in February 2027. So the honest status is not banned, not approved, it's just pending. Unbanned is not the same as endorsement. Removal from a list of safety concerns is a statement about the strength of the evidence behind that concern. It is not an affirmative finding that something is safe or that it works. Those are different findings, and the agency did not make the second one. Anybody selling you certainty right now in either direction is selling you something that doesn't exist. I would urge most people to concede the safety concern and then probably reject the framing. An analysis of 6,441 gray market peptide samples across 14 compounds, including BPC-157, TB500, and GLP-1s, applied two quality standards, one approximating what's required of compounded medications, one closer to what's required of FDA-approved peptide drugs. Depending on which standard you use, between 41.6% and 71.1% of samples failed basic quality criteria. Measurable endotoxin contamination was in 15% of them. In 2024, analysis published in JMIR looked at 3 online semaglutide vendors. All 3 advertised 99% purity. Actual semaglutide content ran between 7.7% and 14.3% in one vial. More than 92% of the contents were unidentified compounds. A Belgian forensic analysis of seized injectable peptides found residual industrial synthesis solvent in 100% of samples, arsenic or lead above pharmaceutical safety thresholds in 26% of them. Now, the certificate of analysis, everybody waves one. Almost nobody can read one. The standard COA reports HPLC UV purity, usually 98 or 99%. That number confirms the target peptide is present. But here's what it can't tell you, what the remaining percentage is. It can't detect bacterial endotoxin. It can't detect heavy metals or residual solvents, and it says nothing whatsoever about sterility. On top of that, it comes from a laboratory nobody inspects with no independent verification and no consequence for getting it wrong. So 99% pure is a chemistry claim. Sterile is a manufacturing claim. Two totally different subjects, two totally different texts, and when you're putting something on your skin, you should probably verify both of them. Every one of those failures is a process failure. Synthesis shortcuts, no endotoxin assay, no sterility test, reconstitution in a non-sterile room. And in my experience, one more that gets almost no airtime, people not understanding how fragile these compounds are once they have them, how to reconstitute, how to store, what heat does, what repeated freeze thaw does. A peptide can be manufactured correctly and be compromised by the time it goes in. That's actually why self-reported peptide experiences are so hard to interpret. Somebody says it did nothing. Maybe it did nothing. Maybe it just sat on the porch in the sun too long. So roughly 70% of FDA-registered API manufacturing facilities are outside the United States. India supplies about half the finished generic medications consumed in this country, and India depends on China for something like 70 to 80% of its own active ingredients. The medicine in your cabinet right now, the FDA-approved kind, is already built on that supply chain. So the media encouraging fear of origin needs to acknowledge that the same manufacturing base that makes your approved drugs safe makes your unapproved ones dangerous. At this point, it isn't even a conspiracy. Pharmaceutical advertising is the 3rd largest category of television and streaming ad spend. By some estimate, it accounts for around 20% of all advertising on TV news, concentrated on broadcasts driven by news and daytime programming. Nobody has to make a phone call when one industry is a fifth of the ad revenue of the outlets covering the industry. You just need editors who'd rather not find out the hard way that they did something wrong. Now, none of that means it's a free-for-all. Pharmaceutical companies manipulating a narrative doesn't make the underlying substance proven. More peptides are not proven, and the most popular ones are among the least studied. But if the clinical literature is thin, and it is, it's worth asking where everything people believe about these come from instead, because it didn't come from nowhere and it didn't come from journals. So in 1981, a bodybuilder named Dan Duchesne wrote an 18-page pamphlet called The Underground Steroid Handbook. He built it out of anecdotes collected off the floor of Gold's Gym in Venice, advertised it in fitness magazines, martial arts magazines, and Soldier of Fortune. He sold something like 40 or 50,000 copies at $6 a pop. Before it, a lifter learned dosing from another lifter, occasionally a sympathetic doctor or out of a medical reference book, where every dosage listed was written for anemia and muscle wasting. The information existed. It was answering a different question, though. Anabolic steroids were pharmaceutical products first. They were developed by drug companies. What came from the underground wasn't the molecules. It was everything anyone actually knows about using them. The doses, the timelines, the side effect management. That knowledge was built by lifters testing on themselves and each other and writing it down for the next guy. Researchers have a name for it: virtual enthopharmacology, literally the pharmacology of the people developed from inside that people. In 1996, 15 years after Duchesne's pamphlet, and decades after gyms had settled the matter, the New England Journal of Medicine published Bason's trial. 43 men, 600 milligrams of testosterone enthanate a week, 10 weeks. The paper's own opening framing: athletes take these substances to increase strength, and the efficacy for that purpose is, quote unquote, unsubstantiated. The editorial that ran alongside it conceded outright. Athletes had been saying for years that androgens increase muscle mass, and it took until 1996 for a clinical trial to demonstrate it. The gyms were right, and they were right first by about 4 decades, which tells you something about how this actually works. The demand almost always shows up before the research does. Pharmaceutical companies are businesses. Something has to look effective before it looks profitable enough to study. But forum knowledge is an N of 1. It's controlled and unblinded and structurally biased towards the people who had good outcomes and stuck around to post about them. The ones who got like 0 results or were hurt left or didn't post. And the threads where the products get reviewed are frequently moderated by people who sell those products. There's a study of DMP users, an industrial chemical, and it's in gyms because Duchesne pulled it back out of the Stanford trial from the 1930s, which tells you exactly what the error bars on this method look like. That study found users made conscious, informed decisions, prepared for side effects, and often took steps to verify the substance was genuine, while maintaining what the research called a nonchalant attitude towards experimenting on themselves. So people should be careful about the vial. It's a personal freedom to be casual about what you put in your body, though. So look at what those communities built on their own with nobody telling them to. One of the largest steroid forums online runs a dedicated subforum for posting full lab panels, blood work, reference ranges, contacts tracked over years, read by other users looking for patterns. They got there because self-experimentation demands it, and they knew the medical industry was never going to develop the kind of substances that they felt like they needed. If you're going to be the study, you also have to be the instrument. So here's where I land on all of this stuff. They were right about the mechanism and wrong about the method. They found real effects a generation before the literature did, and they found that by taking the substance and watching what happened. That's not nothing. It's also not enough to make it a commercial product. So it's not a problem to experiment. It's probably a problem not to measure. So let's take the most popular peptide as of late. If somebody tears something, an elbow, Achilles, shoulder, whatever, within about a day, they've read that BPC 157 accelerates tissue healing. Within about a week, they probably have a vial sitting on their kitchen counter. And all of that happens before anyone has asked what caused the injury, how long that tissue actually needs, or what loading protocol is going to rebuild it. But here's the state of the evidence. BPC 157 has no published control human trials. There's one small open label pilot and a substantial body of animal work. That's about it. Now, set that next to the alternative, honestly. There's a living systematic review and network meta-analysis of lower limb tendinopathies that asked whether any adjunct, injections, shockwave, whatever's on the menu, outperformed exercise, either on its own or added on top of it. It found no convincing evidence that any of them did. The recommendation was exercise monotherapy as first-line treatment for Achilles, patellar, and gluteal tendinopathy for at least 3 months before any adjunct is even considered. So the comparison people think they're making is peptide versus nothing. It isn't. It's peptide versus the best supported intervention in the entire field, which they're skipping in order to afford the peptide. And there's a structural problem no injection touches, and that is to recognize that a tissue failed under a load it couldn't handle. That happened for reasons having to do with training stimulus or load tolerance, how you move, what you loaded, how fast you added it, what you did the last time this happened. No substance you introduce into the body can overwrite tissue that's been improperly cared for. You can potentially accelerate a healing curve. You cannot inject a change into the thing that caused the tear. If nothing changes about how you load and how you move, that tissue fails again, later, maybe, somewhere adjacent, maybe. Damaged tissue needs information to heal. That information comes in the form of stress through movement in a dose that you can tolerate and recover from. The peptide teaches you nothing. Best case, it buys you time that you then spend not learning. Peptides sit at the end of a very long list of things with far more predictable results, and almost everyone who asks me about them is trying to buy their way past the middle of that list. So the standard defense of peptides is that we just don't have enough data yet, and it's fair enough. So let me show you an intervention in the same category where the data does exist and where the objections I've been making about sourcing are structurally impossible. Platelet-rich plasma. They draw your blood, spin it, concentrate the platelets, inject it back into the injury. The RESTORE trial, published in JAMA, compared PRP against saline injections for knee osteoarthritis over 12 months. It found no significant difference in symptoms or joint structure. One of the co-authors described PRP as widely used and typically expensive and ineffective compared to an inert comparator. A randomized trial in patellar tendinopathy prepared leukocyte-rich PRP, leukocyte-poor PRP, and saline all on top of a rehab program. Neither PRP formulation beat saline. A 3-arm double-blind trial in knee OA. PRP, plain plasma, and saline found no difference at 24 weeks, and the PRP group actually had a higher rate of adverse effects, mostly transient increases in pain. The editorial that ran alongside RESTORE noted similar null findings in ankle osteoarthritis and Achilles tendinitis. And the way it put it, it was that part of holding on to participants who got PRP improved substantially. They just didn't improve more than the people who got the placebo. PRP does beat hyaluronic acid in several meta-analyses. It doesn't beat saline in the best placebo-controlled trials. Both of those are true at the same time, which means whoever picks the comparator picks the answer. Better than the other injection and better than the needle by itself are completely different claims. They get sold kind of as the same thing. And now put PRP next to everything I said in the previous section. PRP is autologous. It's your own blood. There's no sourcing question, no purity question, no endotoxin question, no China question. Every single objection people raise about peptides is structurally impossible here, and it still doesn't clearly beat saltwater, which should tell you the sourcing panic was never the real issue. The real issue is whether the intervention does anything, for whom, and compared to what. Let's talk about the actual risk, not the Chinese boogeyman, and not the threat these things pose to a drug company's revenue. Peptides are immunologically foreign amino acid sequences. Your immune system can generate antibodies against them. This is documented in FDA-approved pharmaceutical-grade peptide drugs like GLP-1 receptor agonists can provoke anti-drug antibody formation, and the documented consequences run from injection site reactions to neutralizing the drug's effects all the way to anaphylaxis. That's the approved, regulated, pharmaceutical-grade class. The immune system doesn't care about the certificate of analysis. There's a second pathway that matters more for the people I work with. A lot of reactions to injected peptides aren't classical IgE allergy at all. They're mast cell responses mediated by histamine, and how reactive your mast cells are on a given day isn't a fixed trait. It depends on how much inflammation you're carrying, whether your gut barrier is compromised, how efficiently you're clearing histamine, your hormonal status, your nutrient status. DAO, the enzyme that clears histamine, depends on cofactors people are commonly short on, and its function is impaired by gut inflammation and by a number of common medications. The DAO and mast cell mechanism is biologically coherent, and it's clinically observed. It has not been formally validated as a predictor of peptide reactions. I'm telling you what we watch and why we watch it. I'm not telling you that it's a clear safety net. But the practical implication holds regardless of how that mechanism eventually shakes out. A reaction is not only a property of the substance, it's a property of the interaction between that substance and the state of your body on that particular day, which is why two people can inject identical vials from an identical batch and have completely different weeks. There's another thing that catches quite a few people off guard, which is the timing. Anti-drug antibodies develop with repeated exposure, which means somebody tolerates doses 1, 2, and 3, concludes they're fine, and then reacts on the 8th or 9th. Then they're lying there with a swelling throat trying to decide when the appropriate moment to drive to the ER is. Tolerating the first several doses tells you very little about the next one. And sensitization can be durable. You don't get to choose whether it is. That asymmetry is the entire argument for slowing down. The upside of rushing is a few weeks. The downside is a response you may carry to a compound you might genuinely need later. So you are going to be an experiment either way, whether these things are approved or regulated or not. Every intervention you have ever run on yourself was an experiment. Every training block, every diet, every supplement, every substance. The reason we test before anything is simpler than accuracy anyway. It's attribution. If you inject something and you feel worse 3 weeks later and you have no baseline, you cannot separate the peptide from whatever was already happening in your body. You'll guess, and you'll guess in whichever direction you were already leaning because it's a hunch. Testing first isn't primarily about diagnosis. It's about being able to assign a change to a clause. In the version of this that costs people the most, waiting till something alarms you means the first number you ever see is the broken or deficient one. You have no idea whether it's been that way for 10 years or it just showed up last spring. A set point is worth more than the substance. It appreciates. Every panel after it becomes readable because of it, and the vial that you buy on the market depreciates the moment you use it. So ironically, people will agonize for weeks over the cost of testing or a panel, the cost of knowing themselves, and then hand over the same amount of money without blinking for something that might fix a problem they never confirmed they had because of somebody that they saw said something on TikTok. Some of our own clients do this. They come in for the peptide and want to skip everything that makes the peptide work. And if you think the answer to all of this is just wait for better evidence, I want to show you what happens when the evidence arrives. Testosterone replacement, for about a decade, it carried an FDA warning and a cardiovascular cloud over it based on conflicting observational data. Then the Traverse trial, over 5,000 men, hypogonadal, with elevated cardiovascular risk, followed a median of roughly 33 months. Testosterone was non-inferior to placebo on major adverse cardiac events. No increase in prostate cancer. It's the largest and best evidence we have, and it substantially settled the cardiovascular question, which was good news. It was real news. Now read what people who ran it said about it. The study chair Stephen Nyssen at the Cleveland Clinic went out of his way to say the findings apply to that population and not to the men who have no indication for testosterone but receive it anyway through low T centers. His words were that the study should not be used as a justification for widespread prescription of testosterone to aging men. A safety finding in an indicated population is not a permission slip for an unindicated one. Better evidence didn't remove the indication question, it sharpens it. Lower number and you feel fine, that's a reason to wait and watch. Low number and you're symptomatic, now you have something to discuss with a practitioner. That's the first gate, and it applies whether the intervention has one small pilot behind it or 5,000 patients. So this follows a similar arc to peptides. TRT went from regulatory warnings and declining prescriptions to a landmark trial to direct-to-consumer clinics prescribing off a single low reading and a symptom checklist. The evidence got better, the practice got worse. Nothing about a substance becoming better studied protects you from it being handed to the wrong person for the wrong reason. So this is why the criteria for our prescription is dense. You need all four. Failing one of these is a no from us. The first one is indication. Is there a mechanism, plausible target here, and has the higher yield intervention already been run to completion? For soft tissue, that means a full progressive loading protocol. For fatigue, it means sleep and training load were genuinely addressed, not gestured at. For weight loss, it's a structured way of eating that can reduce the harm of GLPs. The second one is the terrain. Is the system currently able to receive a foreign sequence without overreacting? Inflammatory load, gut barrier, histamine handling, hormonal context, some signs that can help that don't involve formal testing. How bad is your hay fever? Do you find yourself becoming more intolerant of foods or less? The third one is source, a legal pathway with sterility testing and endotoxin testing, not just a purity number from a lab nobody audits. And if a provider is offering you a backdoor to great market product, they've just told you their risk tolerance. That's the most useful thing you could learn about them. They don't care about your health. The fourth and final is monitoring. A defined endpoint, defined stop conditions, and a decision made in advance about what a reaction means, written down before the first dose, not improvised at 10 p.m. during the reaction. Peptides are not a scam. Some of them are going to matter a great deal. The people in gyms and on forums who have found real effects decades before the literature caught up were not idiots, and the reflex to dismiss them has been wrong often enough that nobody should probably join that, but they're a tool at the end of the list, not a shortcut past it. And the two loudest voices in your ear right now, the company protecting 12 years of exclusivity and the vendor shipping unlabeled powder, are both making a commercial argument dressed up as a clinical one. Politics is the worst available filter for deciding what goes into your body because it's aimed at how you feel about a thing rather than what that thing does to you. It made a virus's origin unspeakable in 2020 and a country of manufacture damning in 2026. It kept a compound with no established lethal dose in Schedule 1 for 50 years and then started moving it the month it became ownable. And it'll do it again next year to something else. So this was never an argument about whether peptides are good or bad. It's a way to look at the question of whether they're useful to you or a waste of your money and your immune system. So test first. Fix the structural thing, and then maybe if all of this stuff aligns, try the peptide in that order or don't even bother. Last time, none of this is medical advice, and none of it is a recommendation to start or stop anything. If you want to actually work through your own situation, talk to a licensed provider who can look at your history. As an aside to all of this, there isn't a substance in this podcast that I have talked about that I haven't personally tried. And yes, that includes a lot. That doesn't mean that I was not careful about it. It also means that sometimes when I would try a product, it came from a questionable source. And then we learned the difficult way from bad reactions and maybe even just stalled results or imperceptible results that the source mattered. The reason I ranked all of these things together is because there really isn't a good source of information out there that you can trust. And I'm not saying that you should trust me. That's why I don't tell you whether to do them or not. I'm not telling you to partake in peptides or to try psychedelic therapy because it's not for me to say. But a lot of these substances I have tried because there aren't good answers out there. I did cover the baseline, generally speaking, before I apply them on top. And that is probably why I have a higher likelihood of success with them. I still wouldn't consider myself a peptide guy. I'm fairly conservative when it comes to injectables because, to be honest, I'm fucking lazy. I don't like injecting things. I don't like having to rely on a product or a source to feel a certain way. So take all of that with a grain of salt. And I hope this helps kind of clarify, at least illuminate some of what the argument that's happening right now.